So thank you very much for having me again for this second part of my lecture at the Hyperscanning Summer School. And so last week we looked at theory and experimental design for fNIRS hyperscanning experiments. And this time we will focus a little bit more on practical insights for analyzing fNIRS hyperscanning datasets. Now, some of these considerations, these insights will be more general. They will be also relevant for doing single-person imaging, because we first have to, of course, get the data from each individual by themselves. But some considerations will also be specifically relevant for hyperscanning. And of course, even though I would love to, it's not really that easy to mention everything that is relevant within just a 1 hour lecture, but I will give you some resources at the end, and hopefully we can also stay in touch and answer some questions if you should have any. But let's hope that today I can give you a general overview of some of the considerations that would be good to keep in mind when analyzing fNIRS hyperscanning datasets. So after talking about the general principles of fNIRS, what the role of interpersonal neural synchrony might be, and looking at designing a good fNIRS hyperscanning study, we will now turn to data collection and data analysis. And so when we are collecting data, there are a few things that we could or should take into consideration. Some of them are rather basic, and I'll cover them very quickly because they have probably already been mentioned in some other lectures as part of this summer school. And the first question, of course, is why should we use fNIRS and not any other neuroimaging modality? And here we can say that fNIRS has a few quite important advantages, right? So, for example, in comparison to EEG, it has a good spatial resolution. We know quite well where the signal is coming from, namely from the brain area in the middle of where the channel is, so in the middle of an emitter and the receiver. And I will show you also a tool that we can use to quite precisely determine where we actually want to measure the data from. The temporal resolution of fNIRS is also okay. We can nowadays sample with up to 50 Hertz, but of course, what we actually get in the end is still dependent on the volt signal, and that takes a couple of seconds. So even though we have a very high sampling rate, we still can't really look at too fast neural processes. One of the major advantages, especially if we are also working with children, or if we want to have very naturalistic, very ecologically valid paradigms, is that fNIRS, of course, is less prone to movement artifacts by itself. And also, there are quite a few robust methods to get rid of various artifacts related to motion, but also other physiological signals. And we will mention some of them today. And finally, it's also quite efficient, right? So these days you can pre-arrange the optodes on the cap. You can put the cap on, and then you can just do some data quality checks without having to put any gel, or afterwards also you don't have to wash the participant's hair. So it's also quite efficient in terms of just how much time you need to run an experiment. Of course, we also have to be aware of a few disadvantages or drawbacks. One, of course, is that we can only measure signal from the cortical surface. As I already said, we can't really measure very fast neural processes, like in the millisecond range, like with EEG, because we have the dependency on the volt signal. And depending on the frequency of interest, we need quite long task durations. And we already touched on this last time, and I will explain a little bit more about this also today, which is, of course, important for also designing the task and for extracting the data in a meaningful way. But overall, I think the advantages clearly outweigh the disadvantages, especially if we are aware of some of the limitations and we take the right measures to mitigate them already from planning the study, but also then when analyzing the data. In terms of spatial resolution, as I said before, we know quite well where the data is coming from. where the signal is coming from, because it's always in the middle between the emitter and the detector. Usually in adults, this is about 3 centimeters. In children and infants, it's a little bit less. It's about 2.3 centimeters. This means that we can look into the brain a maximum of maybe 2.5 to 3 centimeters. So we can capture everything that is happening on the brain surface up to a depth of about 3 centimeters. And we, of course, have to then be aware where we actually want to measure from, because it's quite important, because we are measuring really just snippets between our channels. And for that, most of the fNIRS hyperscanning studies that I have come across lately use optode arrangements that either look at the prefrontal cortex, medial, orbitofrontal, dorsolateral, or more anterior regions, temporal regions, like, for example, especially the temporoparietal junction. And so the first thing that you need to do is, when you plan your data collection, is to design an optode array that captures the activity from where you actually want to measure, that is related to the processes that you want to assess. And so the first question is, can I get to those regions? And if so, how can I design the template, the optode template, to measure as many channels as possible? And this is, you can do this in different ways. And so the first way to do that is to refer to pre-made templates. And here is a little video of a pre-made 27-channel prefrontal cortex template provided by Artinis, where you can select the 2D grid in the analysis software, and then you can translate it directly onto the cap. Of course, this looks a little bit different because in 3D there is some curvature, and so it's not exactly the same way as it is on paper on the screen. And there are many different templates available, also depending on how many channels you have and where you want to measure activity from the brain. However, sometimes you want to have a more individualistic template, where you want to specifically select certain regions of interest, and then it's better if you use a free template. So you will purchase caps that are not pre-punched, that don't have any optode holes for the optodes, but you will then punch them by yourself. And one way of guiding where you want to punch them, besides using the 1020 EEG locations, is this very useful fold toolbox. So fNIRS Optodes Location Decider toolbox that has been published a couple of years ago, where you can put in or look at where you want to measure from, and you can also determine how many channels you have. And then it gives you an idea of how you could optimally arrange these channels on your cap. And yeah, this works pretty well, and this is freely available. So I would encourage you to have a look at that and see whether this can help you guide your data collection in terms of where you will put your optodes, if you don't want to stick to pre-made templates and pre-punched caps. Finally, of course, we need to take into consideration that depending on the cap size, which is of course representative of the head size of the participants, we will have more or less available holes for placing the optodes. And so depending on the age range of your participants, you should also take that into consideration, especially if you work with young infants, so that you can have most comparable optode arrangements for the infants. And if you, for example, want to scan them together with the parents, for the adult parents, the adult participants as well, so you can then really look at coherent or matching synchrony between the same regions in the different brains. Another consideration for data collection is, what are we actually collecting, and how do we compare what we are collecting? And here we are talking about baseline tasks or control tasks. And this is quite important. And here there has been a bit of a development over the last decade or so. So initially, in the first hyperscanning studies using fNIRS that have been conducted about 10 or so years ago, mostly the comparison was done between one condition that you wanted to actively investigate and then a rest control condition. And so the rest control condition was not related to the task, and the brain activity during that condition was representing a resting state where you would not expect too many changes in cerebral blood flow. And this was sometimes also referred to as a baseline measurement. What is important here, of course, is that if people come from the EEG field and transition into fNIRS hyperscanning, is the fact that in fNIRS hyperscanning, as we usually also do for fMRI, we don't really have a baseline, right? So we don't, for example, remove some data before an event from our event-related potential, for example. So that's not, we are not really using baseline in that sense. And also, as I have already said last week, we don't exactly know what is going on during the resting conditions when participants, for example, are told to sit back, relax, and don't do anything, but they can actually do many, many things. And so nowadays we are recommending that we don't only have a baseline measurement, even though sometimes it's necessary. For example, if we also want to look at heart rate individually, or heart rate coherence, synchrony, where we need a resting baseline before we start our paradigm. And so it's perfectly fine to also have some fNIRS data from that resting baseline condition. But usually what we are recommending is that we use an active control condition that is related to some aspects of our task that we actually want to investigate, and that represents brain activity as close to the experimental condition of interest as possible, with some important differences, and those differences would ideally be the ones that we actually want to investigate. So for example, as here in the picture, we had a puzzle solving task, and we had the active condition of interest where both participants had one puzzle that they solved together, which we called cooperation, and in another is shown in red. And in our control, in our active control condition, we had both participants having a puzzle each, and they were separated by a removable barrier, and they were puzzling by themselves. So we have all the visual, tactile aspects of the task in both conditions, but what is really different and what we want to get at in the cooperation condition is this social interaction, the co-creation of something. And so this would be a good active control condition, and we encourage researchers to do that. Of course, sometimes active control conditions are not possible, right? If we have very strongly naturalistic paradigms, and we already talked about this last week, and there we recommend to do some permutation analysis, some diet scrambling, or some phase scrambling, and I will get to that towards the end of my today's talk. So the second thing that we talked about already a little bit last time, and that I want to expand on today, is task or condition duration. And as we have seen last week, we have this 2 to 3 oscillations rule, where for reliable WTC estimation, Waveful transform coherence estimation, there need to be at least 2 to 3 full oscillations at the assessed frequency. And this does not only create this cone-like structure, this cone of interest of the WTC data itself, but it also predetermines the minimum and condition duration. And so if we, for example, want to have a minimum, if we only want to look at a task duration of 30 seconds, then the lowest frequency that we can be looking at is about 15 to 16 period seconds, which of course limits the frequency range that we can look at, and that we should actually also be analyzing. And if we then also want to analyze 30-second epochs that are alternating in task content, for example, then this the data that we can actually look at is even further decreased. And for that, I would like to refer to a very recent study published by Catherine Kostorz and colleagues that did a very interesting analysis, namely to look at short windows of interbrain synchrony and how reliable the WTC data is that we can actually get from such short windows of interbrain synchrony. And what the researchers did is that they had simulated data, they had real data, and they really looked at different lengths of signal from 50 seconds to 100 seconds. And what they found is that within that range of task or condition duration, the WTC data was actually comparable. And what I just showed you before, even if we go down to 30 seconds, we actually have quite reliable data at the frequency bands that we can look at. So here, for example, they always looked at a frequency band, I think, between 5 and 6 period seconds up to 15 period seconds. And what you can see here also from the panel C is the importance of considering the cone of interest, right? So if you have, let's say, a task duration of 50 seconds, first of all, you can only look at a reduced frequency range, but within your 50 seconds, you will see that you always have a cone of interest, and that cone of interest grows in size in the sense that we have to remove more and more data at the beginning and at the end of these 50 seconds, the lower we go in the frequency, so that in fact the resulting task window shown, for example, in the rectangle is becoming, of course, also quite narrow. So we don't only lose frequencies, but we also lose a bit of data at the beginning and at the end. But taking all of this into consideration, if we have task durations of 30 seconds or 40 seconds, or 50 seconds to 100 seconds, that doesn't seem to make too much of a difference in terms of the wavelet transform coherence data that we actually can extract. So our task or condition duration can be relatively short, but still it shouldn't be less than 30 seconds. Usually it is within the range of 1 or 2 minutes, but it can, of course, also be longer. So this is just something that you can take into consideration when you plan your study. You should think of at least a task duration of 30 seconds, maybe ideally a minute to 2. And so you have to think rather in a block than in event-related design that you might be accustomed to from EEG studies or jittered rapid fMRI paradigms. Right. The next thing that is really important, of course, when we are collecting data is signal quality. And as I've said before, for fNIRS, there are quite a few means to correct poor signal quality, and I will touch on some of them in just a minute. But of course, what we want to have is good signal quality from the very beginning. And because, especially if we do hyperscanning, if one channel has bad quality in one participant, then we, of course, also lose the same channel from the other participant or the other participants, even though in them or those, this one channel would actually be good and could be used in individual analysis. So losing channels or removing channels can quite quickly escalate, because if you have 1 or 2 channels that are bad in one participant and 1 or 2 bad channels in the other participant, but these channels are not the same, then for the whole dyad, you already have 4 channels that you have to remove. I won't go into too much detail regarding what you can do if you have not that good data, because there are many resources on the Artinius website, for example, of course, as well. But many times it has to do something with the hair interfering with the signal, that you have optode cables that are not properly arranged, that you have too much light, external light, that you can quite easily amend by closing the blinds. You can also, if you go outside, you can buy an overcap or design an overcap that absorbs some of the natural light. We will, these days, we also consider or recommend to use short channels, and I will talk about this later today as well. The good thing is that with the newest devices, you can adjust optode distance and light intensity, so you can play with these parameters. before you start data acquisition. And you can also look at gain, for example, although there are quite a few different ways of determining signal quality at the beginning. And that brings me, of course, to the next question. How does good or bad data actually look like when you start your data acquisition? And here you can rely on the software package that, for example, is provided by fNIRS, that indicates just very simply, you know, green is good data, orange is medium data, and red is bad data. But you can also go to a little bit more into detail and look at your raw data, your raw signal, which we usually do, for example, in our lab. And here, what you want to see, shown on the top panel in the figure, is that you don't have too much fluctuation in your deoxyhemoglobin data, shown in blue, but that you have a clear heartbeat in the oxyhemoglobin data, and that this heartbeat is in the expected frequency range, so maybe half a hertz to a hertz or so, depending on how nervous your participants or how old your participants are. What you conversely don't really want to have is a rapid and inconsistent variation in both the oxy and the deoxyhemoglobin signal, not particularly responding to the heartbeat, which you see in the bottom panel. And you also don't really want to have equal amplitude and opposite signals in those two time graphs for the oxy and the deoxyhemoglobin. So this is also quite an easy indication when you look at the raw data while you arrange the caps and the optodes on the caps in your participants before data acquisition. What we also don't really want to have in our data are rapid, large motion artifacts, even though these shouldn't be too common with the modern devices. Of course, this can happen more likely if you have children as participants, if your participants touch the caps, the optodes, if they move really, really rapidly. But you don't also want to have, you know, huge spikes or baseline shifts or anything like that, even though you can quite nicely amend these when you do the data pre-processing. But of course, the best thing is not to have them in your data from the beginning. So I think these are some of the most important considerations for data collection. Some of them are quite basic, and others are a little bit more advanced, but I think that they make lots of sense, and that you should always try to have the optimal optode arrangement and the optimal data quality before you even start to obtaining the data during your tasks of interest. But once this is sorted and you have collected all the data, the big question then, of course, is how do I actually pre-process my data? How do I make it ready for analysis so I can look at interpersonal neural synchrony or coherence in my data? And here, a couple of years ago, together with Chin and Stephanie, we published a guide to parent-child fNIRS hyperscanning data processing and analysis. So even though we say that it's for parent-child, it's also completely viable for adult-adult hyperscanning. And in that guide, we present a flowchart of all the different steps that we think should be done in order to have the best possible data quality for your interpersonal neural synchrony analysis, which is at the very bottom of the flowchart. And as you can see, this involves quite a few steps. And I won't be able to mention all of these steps today, but I will go through some of them and give you some recommendations. And what is also quite neat is that this guide to parent-child fNIRS hyperscanning data analysis also comes with some sample data from 20 parent-child dyads. And this sample data is uploaded on OSF. And the sample data is not just there in its raw form, so without any pre-processing done, but it's actually also there after pre-processing. So you can compare what we get when we pre-process the data and what you get when you hopefully use the same pipeline. And what also is there are all the MATLAB scripts that you need to actually do those pre-processing steps. Now, one thing to say is that when we published this data, we had obtained the data from using fNIRS devices. So some of this code is specific to that. However, we just recently had another fNIRS hyperscanning workshop at Essex, where we actually analyzed Artinius data. And so we amended this pipeline to also be compatible with Artinius data. The differences are minimal. The steps are exactly the same. It's just that the data structure is slightly different. And so please get in touch with us if you would learn a little bit more about that, or if you want to have access to some of those scripts. We are still refining them, but hopefully not too far in the future, we might also be able to make them publicly available. But in the meantime, you can maybe play with this. And if you have a specific need to have it also compatible with other manufacturers' devices, then we can certainly provide that. Yes, one thing that I forgot to say, on the OSF page, we also have the R scripts available, because once we derive the WTC data, we then take all of that data and analyze it in R. And so you have all the R scripts available as well. We think that R is particularly well suited because you can nest data. So for example, you can look at ROIs, but still have the individual channels in there. And also you can build quite complicated models that can take into consideration some missingness, right? So if from one dyad you have one channel missing, and from another dyad you have another channel missing, you can still use all of the dyads, because R can handle this missingness quite well. So please feel free to check this out. I will show at the end of the presentation where all this information is. Have a look, try to work through the pipeline to see what all of these different steps do, because as I said today, I can only give you a very brief overview of some of these steps. Of course, there are different ways of doing the pre-processing. So our pipeline takes elements from quite a few different software packages. For example, Homer2 in MATLAB, SPM for fNIRS in MATLAB, Brain Analyzer in MATLAB. But you could also use MNE in Python, or you could use Artinius own pre-processing pipeline. So there are quite a few different ways of doing this. We like to use these MATLAB scripts because each pre-processing step has a separate little script where we can also play around with the parameters, so we know exactly what goes in and why we get a certain result, which is sometimes not as intuitive. And also you can, of course, do everything step by step and actually see what happens to your data. And so the basic steps that should be done is, or that what we recommend, is a data quality check, and I will show you various means of doing that. Then we have to convert the data to optical density. We have to do a motion correction, or physiological artifact removal. Then we usually do some bandpass filtering to also get rid of more physiological noise, and then we convert to changes in oxygenated and deoxygenated hemoglobin, and then we do the wavelet transform coherence analysis. And so one thing that we think is really, really helpful is that very soon after you have acquired the data and before you do any fancy automated analysis, that you actually do a manual visual data check. And this is really simple, because all you have to do is that for each participant, you just have to derive a time by frequency plot for each channel. And once you have done this, you look for the heart band, which is basically the band around here in this data, around 0.5 hertz, because this is data from children. And what you want to see is a clear, continuous, uninterrupted, not too motion artifact covered heart band. So channels on the left side, 11, 12, 5, 15, and 16, show a very nice heart band at the top at the 0.5 hertz or period second range. Whereas the channels on the right, channel 4, 11, 8, and 15, do not show that, right? So in channel 4 and channel 8, we don't have a heart band at all. In channel 11, something went quite wrong because the data looks really strange. And in channel 15, we do have a heart band, but the heart band is quite covered with these spikes, which from the lower frequencies, which points to quite a lot of motion artifacts. Of course, we are aware that this is a manual visual data quality check. that it cannot be really reliably automated. And if you are acquiring lots of data from lots of participants, this might be a little bit tedious. At the same time, we recommend checking your data quality after each acquisition to make sure that you don't have any problems that are persistent and that you don't acquire 20 dyads and then you see that in all of them you have some technical difficulties and you have to discard the data. When we do this in our scripts, we also have it automated that we see all the channels from each participant on one screen, because we usually have about 16 channels or 18 channels. And so it's really easy to do this quite quickly, to note down which channels you would remove from this visual data quality check and then move to the next participant and then move to the next dyad. So quite simple, quite straightforward, and that we would really recommend doing this visual data quality check. Now, of course, there are also some more automatic data quality checks that you can do on top of this, or that you can just do as an alternative. One of them is an automatic labeling of bad channels in Homer, which is called prune channel function, which mainly looks at the expected data range, the signal to noise ratio threshold, and the inter-opto distance threshold. And this is something that you can do on top of the visual data quality check. And I won't say too much about this because there is a very nice video about that, how it works, how you can input all of the things, and how the result looks like. So this is one automatic data pruning option. The other option that quite a few labs are using is the skull coupling index, or the SCI. And what is done here is that from each channel, from each participant, we are only extracting the cardiac signal band, so the signal between 0.5 and 2.5 hertz. And then for both the oxygenated and the deoxygenated hemoglobin, or the two wavelengths. And then there is a cross correlation at zero lag computed across these two, the two signals at the two wavelengths. And if we have a higher SCI, this indicates that the two signals are have a very similar time course, and that there is a good scalp contact and good signal quality. And while this usually works pretty well, and you can also adjust the threshold a little bit of the SCI, so I don't know, 75% or 80%, the risk here is that you might get a very high SCI value if both signals that you extracted in the cardiac signal band show exactly the same noise contamination. And so it could be that according to the SCI, you have a very good channel, whereas if you actually look at the visual data quality check, you have something like in channel 15 on the bottom right, where you actually have a not such good data because you have lots of motion artifacts. But you only see this when you visually look at the data, because these motion artifacts could be in both wavelengths, and then you would actually conclude, if you do it automatically, that you have bad data. So what we suggest is that you do both a visual data quality check and maybe a quality check using the SCI. And if you have concordant results, then everything is good. And if you, for example, find that in the visual data something looks bad, but in the SCI it looks acceptable, then maybe you go back and look at the channel a little bit more in detail and discuss it within the group to be sure that whether to include or exclude this channel. So quite a few different options, some more automatic, others more manual. But it's really worth to explore all of them, because you want to make sure that once you have pre-processed your data, that you have the best data possible for your WTC analysis. The next step then is motion correction or noise correction in general. And here, one of the main issues is that you have quite a few different means of doing so. You can have spline interpolation, you can have TPCA, wavelet thresholding, temporal derivative distribution repair, and maybe quite likely a few more. And the thing is that most of those work quite differently, right? So I will explain just on the next slide what the spline interpolation does. But then in the wavelet thresholding or in wavelet-based methods, you are decomposing the data into different frequency bands, and then you remove certain frequencies by assuming that you have more noise in certain frequencies than in other frequencies. But if you have some actually data in some frequencies that are deemed to be noise, then you can remove that data. And then, of course, that's not really what you want. Temporal derivative distribution, you compute the temporal derivative of the data, and then you use that to remove some bad signal. And again, this of course has some different assumptions and might do different things to your data. So we would recommend that you maybe acquire some pilot data and that you use a range of different motion correction methods to really see what they do to your data and whether they only remove noise or whether they also remove some of the data from the frequencies that you actually want to look at, just again to be sure that in the end you have the best data possible. I'm just briefly going to mention the spline interpolation because that's also the method that is used in our guideline paper that is explained there, and that we also have the MATLAB scripts for. So here in panel A, you see that around 40 seconds, you have a huge spike in the data. And this is then in panel B captured by the algorithm because it derives a moving standard deviation. And of course, in that peak area, all of a sudden you have a huge spike in the standard deviation of the data. Then it marks this window in panel C in the raw data. It magnifies it to really look at it in detail, and then it removes the spike and interpolates the data during that time from what the data was before and what the data is afterwards. And then in F, you see the final outcome where this spike was removed and the data just continues more smoothly and is successfully denoised. So this is one way of doing the motion correction, but as I already said before, there are several others that work differently, and so the best way is to try out a few of them and to see which one works best for your data. Here is another figure from our guideline paper where you can see on the top the raw data that hasn't been motion corrected, and then on the bottom you see the data after the motion correction. And you can see that at least the big spikes have been removed, but sometimes you still have some other trends in the data where you, for example, have a bit of a shift or so. So again, look at your data in detail and see what the motion correction does and how your data looks afterwards, and whether you think that whatever you get afterwards is actually suitable and the best quality possible. The next consideration is short channels. And this is not only a consideration for data preprocessing, but it's also a consideration for planning your study, right? Because when you plan your optodes design, you should, of course, already plan in, if you want to use them, short channels. And of course, short channels are channels that are shorter, so that where the emitter and the transmitter are closer to one another. So usually the normal standard range channels, the distance between the emitter and the receiver is 3 centimeters, whereas the distance between the emitter and the receiver in the short channel is 1 centimeter. And the idea here is that when you have a short channel, whatever you measure is not really coming from the brain. So it's not really the signal that you want to look at, but it's a signal that comes from more superficial layers. And if you have any data components in there, they're most likely to be noise. And so if you then basically correlate out or subtract the short channel data from the long channel data, you should get a cleaned time course that has some of the physiological noise removed. There are different ways of arranging short channels for Artinis. It's quite nice because you can you don't lose standard channels if you want to add short channels. because you can just add a different optoad holder that then has also a space for a short channel just beside it, but you can still use that optoad for a standard channel. You can also purchase some additional sets if you want to have more short channels. So you can purchase bundles of, I think, 8 short channels. But in our paradigm, or in our studies right now, we are using our standard 16-channel layout, 4 channels per ROI, and then we add 2 short channels, 1 per brain region on each side. But of course, this can be extended, as I said, by buying some short channel bundles. The idea of having short channels, as I said before, is that you can hopefully get rid of some of the physiological artifacts that you don't want to have in the data. So, and this is a quite nice paper that has looked at this, because it has on the left in panel A, it has the time averaged wavelet transforms for long channels in dark color and short channels in brighter color. And so you can see which of the motion artifacts are actually quite nicely captured by the short channels. And this is especially the case for high frequency noise. So for example, for the cardiac signal, but also for a medium frequency noise. So for the respiration and the Meyer waves, right, which are related to muscle activity or blood pressure. What, however, is important to remember is that the short channels, they don't really capture very low frequency noise that is attributed, for example, to vasodilatation of cerebral vessels or neurovascular coupling. And so there, you can't really get rid of that noise by using short channels. And that's probably also quite important, because that's usually the frequencies that you want to look at when you analyze your WTC data for the tasks of interest. So whereas you can get rid of some of the noise when using short channels, it's not a perfect noise removal tool that doesn't work for all the frequencies with a similar efficiency. What is also important to consider is that you can't just extract, or you shouldn't just extract the short channel data and then just regress it out using a very simple method. So it's actually better if you combine some of the short channel data, that you look at some of the global components of the short channels, and that you model that in a slightly more complicated model, so in a GLM, for example. But this has now been quite nicely, this method has been quite nicely established and further developed. So it shouldn't be too hard to find some guidelines on that. I will also show you a different guideline paper where the researchers actually did that. So you can refer to that, because in our guideline paper that we published a couple of years ago, we did not have any short channels yet. So there are no scripts for that, but we are working on these scripts because we used them in our hyperscanning workshop that we just had about a month ago or so. And as I said, hopefully these scripts will also become available soon. The importance here is that we shouldn't think that short channels are going to help us automatically in any way we use them. There have to be some things considered, like the frequency ranges where it's actually useful, and how the short channel data is actually removed from the standard channel data. Because if this is done in a not so optimal way, you might actually make your signal noisier than before, and that's of course not something that we want to do under any circumstances. What then also needs to be done is to convert the data into HBO and HBR. And that's not so interesting by itself, but what is really interesting, or what is another consideration that you might remember, is whether you are actually reporting both the HBO and the HBR data, or whether you are actually analyzing both the HBO and the HBR data. And here there has been a review paper published a couple of years ago that found that there is very high heterogeneity of chromophore data reporting in fNIRS research, so that some studies only report one chromophore concentration and not really the other. And so what is usually, or what we of course usually do, is to look at the WTC in terms of the HPO data. There are only very few studies where only the HPR data is reported. But there are quite a few studies where only the HPO data is reported. And I think that what has been happening in the field more recently is that we are asking more and more often to also report the HPR data, to actually also do all the analysis steps that we do with the HPO data, also with the HPR data, at least in the supplement. So this is also there and can be looked at. Because we of course know that it's more likely that what we want to look for is represented in the HPO data, that we have probably some higher signal-to-noise ratio and more sensitivity to task-evoked changes, and that there probably is a closer correlation with the fMRI bold response. But very often there is no real justification provided why people are only looking at HPO data. And so I think that, as I said before, the field is moving to the direction that we should always report both of them, at least the HPR data also in the supplement, and that it's always nice to see also the full analysis done with the HPR data. And yeah, quite often only some findings are in the HPO and not in the HPR. Okay, so I'm slowly getting to the end of today's presentation, where I want to mention a few more steps that are related to the data analysis. So when all the pre-processing is done and we are actually deriving the wavelet transform coherence data, and we want to look for interpersonal neural synchrony or interbrain synchrony, that is the main outcome for our studies. And so one consideration is which frequency or frequencies of interest should I be looking at? And we already covered this a little bit last week, because for certain tasks that are quite controlled, we know quite well what our task frequency is or what our task frequencies are. But for other tasks that are highly naturalistic, it's really difficult to say where we will have the highest power and then probably also the highest coherence in our WTC frequency range. And so one way that this can be partially amended is to decompose our data into the different frequencies, what we can see here in the middle figure, and then only look at those frequencies that show the highest coherence for our task. It's certainly fine to do that if we first acquire some pilot data, and then we look into the pilot data to get at those, to determine the best frequency for our task and for our participants. But we just have to be careful that we don't do this post hoc, right? So that we do some cherry picking, that we then only look at those frequencies where we identify that there is the highest coherence, and that we ignore all the other frequencies, because that wouldn't be really a proper scientific approach. What also can be done, and I already mentioned this last week as well, is that we can look at many different frequency bands. So maybe high, middle, low, or maybe even split the frequency bands in much more fine segments, and then look at the coherence in 10, 20, 50 frequency bands. And this is also perfectly okay, but we should then again not only report the data from those frequency bands where we have the highest coherence, we should report it from all the frequency bands, and we then have to correct for the number of frequency bands that we are looked at, so that we have a proper correction for multiple comparisons. Another way is to average over frequencies. what we have usually done in the past. So for short time windows, for example, for 30 seconds of data, we averaged over a frequency range of 6 to 15 or 16 period seconds. If we have longer task durations, we then average over a frequency band range of 10 to 50 period seconds. So that is also perfectly feasible and perfectly okay. So again, different approaches, but we need to be aware of the limitations, especially not to cherry pick only the significant or the best data and not to report all the other data without correcting for multiple comparisons. And one thing that I also mentioned last time is that most of the fNIRS hyperscanning papers so far use wavelet transform coherence, but that there is another method that also is used, albeit not that predominantly, and this other method is correlation. And here I just want to show you some of the very, very important differences. So here on the bottom of this plot, you see what we have already looked at last week and what we have seen this week a lot. So this is first the wavelet transform coherence plot, the time split by time and the frequency. And so you can see that you have quite consistently high wavelet transform coherence in the frequency range between 4 to 16 period seconds. And on the very top, you see the data that leads to that wavelet transform coherence plot. So you can see that in the first minute or so, the data is completely in phase. So you have a very high coherence value. Then around 100 seconds, you start to have a consistent shift where the red data is always a little bit later than the blue data. And so you still have a very high coherence, which is close to 1. Then around 150 seconds, you start to have the red data being shifted more, being more ahead of the blue data. But again, because this shift in the phase is consistent, you still have very high coherence. And finally, from around 200 seconds, you have the data in opposite phase. And again, your wavelet transform coherence value is very high in this one. And the only intervals where the wavelet transform coherence is slightly lower or is getting lost is when you have these transitions between those consistent phase relationships, right? So when the phase relationship shifts from being perfectly in phase to slightly shifted, when it shifts from slightly shifted to be more shifted, and when it shifts from more shifted to be opposite in phase, that's when you have the lowest wavelet transform coherence values. But for all the other scenarios, you are always within the range of 0 to 1, and you're quite high on 1. And so it's quite nice to do some compatibility analysis. Now, what is different when you do correlation is that you can have values between -1 and 1. And these values differ depending on the phase shift. So when the two signals are perfectly in phase, you have a value of 1. When one signal starts to be shifted a little bit more in advance, you are having a decreased value, maybe 0.7 or 0.6. If you then have a shift of 2 seconds, then your value or your correlation value will become 0. And then if you have signals that are opposite in phase, your outcome value will actually be -1. And so what we can see here is that for this correlation analysis, it is crucial to know what the phase relationships between your data are, because they will yield different correlation outcome values. And so usually you have to do these correlations for many, many different phase relationships, and you can't just do it over the whole data. And the results will differ strongly for different phase relationships. And that, of course, adds, again, more comparisons, which you then have to control for. And that's one of the advantages of wavelet transform coherence, that you can do the analysis and you will get comparable coherence values independent of the phase relationships, as long as these phase relationships are consistent over a certain time window. So I think that's the biggest difference, and that's what should be always remembered. And finally, we talked about this towards the beginning. What now if we have a highly ecologically valid task where we cannot really think of an active control condition? And so what we recommend here is that we do a permutation analysis. And as I said already before, there are two different ways of doing that. And one way of doing this is the so-called dyad scrambling. So for example, in this study, we had 140 true parent-child pairs, and we derived the wavelet transform coherence data for those true pairs, which are always shown on the left in this graph. For the red is the cooperation condition, and green is the independent condition, and blue is the resting condition when they were solving these tangram puzzles. And what we then use as a control analysis is that we take each, for example, each parent, and we pair their data with all the children's data except their own. And so we create 139 random pairs per true pair. All of these different pairs did the task, but they didn't do it together. And so we then generate these 139 random pairs per true dyad. We average their wavelet transform coherence, and we then compare this average coherence of the artificial pairs with the true pairs. And what we want to see is that the wavelet transform coherence is significantly higher for the real pairs as compared to the random pairs for all of our experimental conditions. And if this is the case, we can then go on and only analyze our true pairs, because we know that the true pair data is not caused by some random noise in our data. We actually recommend doing this not only if we don't have a viable control condition, but also if we have a good active control condition, because it is a really, really nice additional insurance that we don't have spurious correlations in the data that we actually see as being driven by, for example, two people co-creating something or mutually predicting each other. Now, the second way of doing this is the so-called phase scrambling, which is described in this very nice analytical pipeline paper that was published quite recently by Victoria St. Clair and colleagues, again for 4 to 6 year old children. So this is also a very excellent guideline paper if you all want to have some idea not only about the phase scrambling, but especially also about the short channels, because this guideline paper includes the short channel regression. But back to the scrambling. So here we don't pair each parent with all the children's data except their own. Here we actually take, for example, the parents' data, and we first Fourier transform this real data into the frequency domain. And then we do a phase randomization. So within the parents' data, the phase angles of the frequency components are randomized, while the amplitude information is preserved. And so what this does is it basically effectively scrambles the temporal relationship between different frequencies in the signal. And afterwards, we do an inverse Fourier transform, which gives us a scrambled data that has the same amplitude or power spectrum as the real data, but it has scrambled phase. And if we then do the WTC between this scrambled phase data and the real child data, we should again have lower coherence than if we do the wavelet transform coherence analysis for both real data sets. There isn't really too much of a consensus which method should be used when. Both of these approaches are feasible. What is important for the dyad scrambling is that the data structure of the real dyads and the artificial dyads is as similar as possible. And this is sometimes a little bit difficult if you have naturalistic tasks that sometimes in some dyads take a bit longer or are shorter than in other dyads. or if you have a sequence of different tasks and you have some rest in between, and this rest is not always the same length for all the dyads, because then if you do the dyad scrambling, you will not always be comparing the same condition in the real and in the artificial dyads. So then the phase scrambling might be a bit more implied. But of course, there might also be some drawbacks with the phase scrambling, because WTC analysis, of course, is quite takes into consideration many different phase relationships, and so that might not always work that well either. So maybe again try out both of them, see what the result is of that, and ideally also report both of the analysis in the paper. And maybe I'll have a further look on how this is actually done and how the results look like. But just for you to be aware that these control analyses with the scrambling approach, the permutation approach, are really good and highly recommended, and that there are currently at least 2 different approaches that can be used to do that. Right, so this brings me to the end of today's presentation. If you want to read more about what I said, again, I would like to point out my fNIRS Hyperscanning and Introduction website, where there are also links to our guideline paper, where you can find other recordings, where you can find other references, where you can also find a link to our OSF page, where we have all the sample data and all the scripts. And hopefully quite soon we will have also some more scripts with taking short channels and also the different motion correction algorithms, and hopefully also the different permutation analysis into consideration, so you can have sample scripts for all of them and try these out on your own data and see what works best for you. And with that, I'm at the end. I thank you very much for your attention. Thank you again to Martinez for the kind invitation last week and this week. And yeah, as last week, I'll be happy to take a couple questions before we finish this session. Thank you.